Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Hi everyone, I'm Samuel Miller from the Microbiomics and Culturomics core facility at Oklahoma State University. My research focuses on combining modern molecular biology with traditional anaerobic cultivation methods to discover novel microbes that thrive in the absence of oxygen. Join me on the Finding Genius podcast, where I'll share how combining these approaches sets us up to discover and describe these yet-to-be-cultivated microbes.
Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius podcast. Our guest today is Sam Miller. He's a postdoc in microbiomics and culturomics core facility at Oklahoma State University. So we're going to talk about anaerobic bacteria that are able to live without direct oxygen on them. So again, Sam's an anaerobic microbiologist interested in yet to be discovered bacteria, particularly ones, again, that grow in the absence of oxygen. So welcome, Sam. Thanks for coming.
Yeah, Richard, thank you so much for having me. It's a pleasure to be here.
Yeah, why anaerobic bacteria, not aerobic? Do you just had to pick something, or is there a reason they're more interesting to you?
Well, I do find the anaerobes a bit fascinating, but I think, like most of us, we're products of our environment. And I spent most of my PhD study in anaerobic microbiology labs. And so I've become, over the years, fascinated with this topic.
Okay, so what is your research about right now? Like, what are you studying and where?
Yeah, so I like to tell people that I sort of split my days 50-50 between being a postdoc and delving into certain things, and then also working for the Culturomics core facility here at OSU. And so I'm involved projects in both aspects of that. In particular, things that are probably, like, if I had to give you like a top 3, not that these are necessarily obligate anaerobes, but we're working on isolating some mixed bacteria. They're a fascinating group of bacteria. They're predominantly found in soils. They have some really cool, unusual behaviors. They're predators. They like go out and hunt down prey and kill them and eat them. But on the anaerobic side of things, stuff that I'm working on is describing some and growing some novel anaerobes from some kind of wild places, things like the gastrointestinal tracts of blue whales, kangaroos, ostriches. Like, you pick an animal, I'm probably working with it. And then also on the anaerobic side of the fence, the lab is also sort of split between bacteria and fungi. And so we also deal in what's called the anaerobic gut fungi phylum. It's called Neocalamastica mycota. But we do similar things in there. We try to go seek out novel anaerobic microbes, whether they're bacteria or fungi, grow them, get them on petri plates, and bring them into culture in a lab. So those are the projects that I'm sort of involved in right now.
How do you get them out of the field if would exposure to air kill them, or would it just disrupt it would cause their bacterial machinery to work in a different way? Like, what would happen?
Yeah, that is a fantastic question, Richard. And in short, the oxygen that you and I are enjoying right now could certainly be not kind to the obligate anaerobes that we like to work with in the lab. And so, yeah, when we're collecting, basically as soon as that, like, say, take fecal material, for instance, as soon as it comes out and it's defecated, it's going to start being exposed to oxygen. But the brilliant thing is that if you think about the fecal materials having like layers, right? It's going to take a while for the ox to penetrate sort of into the innermost layer of that fecal material. And so what we try to do, whether like I'm collecting it myself or I've got a student collecting it, or even if it's, say, a farmer out in Kansas collecting something for me, I just try to ask and try to get it as fresh as possible. And then I try to fit as much of it in a collection tube as I can to limit the amount of headspace, the amount of like oxygen and atmosphere that's going to be in that tube. And then, just try to get back to the lab as quickly as possible so we can get it into some special tools and toys that we have to play with things.
But what's when a anaerobic organism is exposed to air, is it like the vice versa? Aerobic organisms, they can survive without air for maybe a minute or two, but then they'll die. Like, what would happen in the reverse?
Yeah. So in short, the answer is yes. Right. Some obligately anaerobic microbes, they have survival strategies, like they're able to produce endospores, say, when they encounter an environment that is unfavorable, such as, perhaps like an oxygen gradient or something like that, like a Clostridium or something of that sort. It would produce an endospore and then sort of wait for times to get better, wait for an anoxic environment to come back to it. But in general, different anaerobic microbes are going to have different oxygen tolerances. Some can tolerate it for a couple of minutes. Some are more sensitive. Some are less sensitive. But in general, exposure to oxygen is going to harm these bacteria and definitely kill them.
So they use to respirate, if not oxygen.
Oh my. So things that essentially aren't oxygen. We can use, things like various nitrogen and sulfur compounds, other sorts of inorganic goodies like that can be used to, facilitate this type of metabolism.
But what's common? Like, I have methanogenic bacteria. I don't know if they produce or consume it, but I would think there's a very limited set of substances that organisms could respirate upon, even if they're anaerobic, or is there just a lot of them?
Well, there are. It's not limited to just one. I would say the common ones are going to be things like probably like nitrate. That would be a very, very common one.
Okay, so you could donate electrons just like oxygen would to use the respiration.
Yeah, totally.
Okay, so nitrate. What about like sulfur compounds, or again methane, or what are some of the other ones?
Well, I can say that I don't deal a whole lot with the methanogens, methanotrophs, and those sort of organisms. But in general, I'd roll with nitrate and also like sulfate. Sulfate would be another good one for those sulfur-reducing bacteria. There's also quite a few archaea that like to be sulfur reducers as well. And so they'll essentially, like you just said, they'll use that sulfate as an electron acceptor. And, they might produce something else as a byproduct like hydrogen sulfide or something like that.
It's just really weird if you think about it. Like when organisms eat or they respirate, it's not like, oxygen is an atom. It's made of, electrons, blah, 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 protons. What are they really consuming? I guess they're consuming the energy that's at just the right level on some of these bonds. That's what it seems like when you eat. You're really like, again, like electron bonds are the currency of food and breath. It's just really weird. That's what we're consuming essentially,
Yeah, I hear what you're saying.
I don't know. Maybe have you thought about that before? Like, what is that? Does that change how you think about, how this works, or it's just strange?
Yeah, it is kind of strange if you start to boil things down to even their most, basic rudimentary or physical units. It does kind of, it's kind of a weird thing to think about. But no, I don't spend too much time thinking about that. I'll leave that to the physicists. I'll let them get down to the nitty-gritty stuff. Thanks.
Well, energetically, the anaerobes, are they still able to produce enough energy to respirate and move and do their thing just as much as the aerobes, or are they in like a slower, less energetic type of respiratory cycle?
Yes. So the short answer is yes. They do some things just a bit differently. They'll potentially things like, say, fermentation, anaerobic respiration. We've already kind of talked about that respiration with like nitrate, sulfate, other things like that. Fermentation, you can kind of think about it as like a less good version of what happens aerobically if we're talking about like producing ATP. But not to get into like the specific amounts of ATP per glucose or something like that. I don't have that right in front of me, but essentially it's quite a bit more to have that happen aerobically in the presence of oxygen than it is to happen, without oxygen. And so they're still able to produce, to go through those sort of metabolic processes and to get energy from food sources. But it is, as you sort of allude to, much slower a process. So if you think about like the growth and development of bacteria, take the poster child bacteria like coli, that everybody loves. We typically measure things like this in what's called like a doubling time. Like how long does it take to make, like a Xerox copy of itself? And so for E. coli under optimal conditions and like a test tube in a lab, it's something like 15, 20 minutes. So it's pretty quick. A lot of the or not, I shouldn't say a lot, but there are definitely anaerobes out there where that doubling time is into like days. Like it's still longer than just 20 minutes because these processes are just so much slower.
We'll do a lot of the anaerobes occupy like micro environments because around, oxygen is all over the place inside a body. For bacteria, at least, that's a huge environment because they're so small. But again, do a lot of anaerobes just tend to occupy micro environments. That doesn't change. Maybe they don't need to be as energetic and they don't move as much. They're more stationary. I don't know.
Yeah, so you do definitely see like specialization with respect to what types of microbes grow where, like say in the human body, for instance, because there are definitely portions that have, say perhaps like a higher concentration of oxygen in certain tissues and lower concentrations in other tissues. And yeah, so definitely they're gonna specialize in areas that they're going to thrive in. So they're gonna seek out, environments they prefer, and that won't be all of them.
Do any of them have the ability to be like, I don't know if you call it biphase, like just to be able to respirate with oxygen without it? Both, or very few creatures cross over?
So we have, if you want to generalize it into three types of microbes, I'd tell you that there's aerobe, like they need oxygen like me and you, or else they're gone. There's the obligate anaerobe that, are gone in the presence of oxygen. And then there's this middle ground where we have what are called facultative anaerobes. And so these are these are organisms that can grow either with or without oxygen. They can do either one. Sort of with oxygen is preferred, again, because they're going to get faster growth. They're going to be generating more energy sort of per molecule of food that they're consuming. But they can also, in the event of them being in a place in the absence of oxygen, they can certainly, switch over and sort of grow in the absence of that molecule.
Okay. Oh, so you need these, do they act as like, diplomats between aerobic and anaerobic colonies? Do they act as like messengers? Do they transit the colonies? And I don't know, do they perform a different function, the ones that are facultative anaerobes?
Do they perform a different function?
Because they can go between the two worlds, essentially, and live in both. Do they perform some kind of messenger function between colonies of bacteria, that normally wouldn't interact or couldn't?
Not sure that I know the answer to that. I wouldn't be comfortable. Yeah, I'm not sure that I know the answer to that. That's an interesting thought. Yeah, generally we just think about, say, facultative anaerobes. Like again, they perform better in oxygenated environments, although they can, live in the absence of oxygen. I kind of like to think about it as thriving versus surviving, right? Like they're going to thrive and then they can also survive if they need to, but they'd much rather be thriving in the presence of oxygen.
All right. So what research questions are you trying to answer right now?
Yeah. So let me see. As part of the core facility that I'm at, we are tasked, amongst other things, with supporting the research of 5 sort of younger tenured assistant professors that all need to do things with respect to like the cultivation of obligate anaerobe. And so part of what I am doing is helping these professors grow specific type of anaerobic bacteria. Like for instance, we've got a professor that is very much interested in a genus bacteria called Bacteroid. He's very interested in particular components of their genomes and things of that sort. So essentially what I was tasked with doing was growing as many Bacteroides as I possibly could from a particular set of samples. And so in his case, he was like, Sam, I want, these Bacteroides and I want them from wastewater treatment plant. And so that's, sort of where I come in and facilitate that. I'll go out and collect the samples, bring them back to the lab, process them, and then start trying to target the cultivation and the growing of this particular type of bacteria. So that's some of the stuff that I do. We also do a lot of, as the name microbiomics sort of implies, a lot of sequencing. So we've got certain professors, again, that we're sort of helping out that they need like high throughput sequencing of particular samples. Like we have a professor that was wanting us to identify about 1,500 different things. And so he sent them to us, and then using, the magic of high throughput next generation sequencing, we were able to give him like an identification of these 1,500 bacteria that he'd grown in his lab. And so that's part of what I do, is sort of helping these professors with their project, sort of as and when they need assistance with either the sequencing, the microbiomic side of things, or whether it's, a more like cultivation-based approach, like, they want to grow something that's perhaps difficult to grow or something of that sort.
On the flip side, why are so many bacteria hard to cultivate? I've heard.
Yeah, the In short, we don't really know. The term that we throw out there, the royal we here, microbiologists, would be microbial dark matter, which is most of the bacteria. So we've sequenced tons and tons and hundreds of thousands of things, in recent decades as sequencing has become more accessible and more cost effective and all those sorts of things. And so we can put together genomes for bacteria that we yet to grow in the lab yet. And I think at the end of the day, it boils down to us not having, just the right set of conditions, just the right set of circumstances for these bacteria to flourish on a plate. And then some of it too is just the relative abundance of these bacteria in the environment. Like they may be there, but it may be, 0.1% of the total population in that particular sample. And so just sort of by default, they're kind of limited already. There's not a lot of there to begin with. And like we talked about at the beginning of the podcast, it might be like sample collection stuff. You might have had bacteria die from point A to point B, en route to the lab to even analyze that. So there's a lot happening there. Yeah, we certainly haven't grown everything that's out there. That's for sure.
Oh, are there like mid-intestinal bacteria or microbiome projects, that you could help with? Because traditionally they're supposed to be very hard to get to, right?
You mean like perhaps somebody's got like a scope or something and they were able to pull a sample like from a pet? Is that what you mean?
Yeah, like I don't know if anyone's been able to do that. It seems like a lot of the bacteria, before the colon in the body, just can't be reached, can't be cultivated, nothing.
Well, I can say that I haven't personally been involved in projects like that you're describing, but I do know that people have worked with samples like that, clinical samples from, whether it's endoscopy type things or other types of samples that are collected in that manner. But yeah, generally we do deal with like fecal material. So like it's already passed through, sort of those physiological barriers through the GI tract and things of that sort. You certainly can go in and perhaps do a more invasive type of sampling to collect a sample from a particular portion of a particular anatomy.
Okay, yeah, I just didn't know if it would be very useful to collaborate with you, for instance, on a lot of these microbiome studies. Again, because you're coming from the anaerobic side, which is a side, I guess, probably not equally represented, amongst bacteriologists.
Yeah, I will say that my PhD advisor, Paul Lawson, he always used to say that we're a dying breed, the anaerobic microbiologist. So yeah, I would say that there are fewer of us. Yeah, with respect to collecting those samples, I'm probably not the guy to help people like actually go and collect them and, those sort of things. But if a person had a sample already and they wanted to, sequence that particular sample, like send it to me and I could extract DNA, that sort of thing, I could certainly facilitate that portion of it, as well as if they were like, hey, we want to try to grow X type of bacteria from it. Do you think you could help us out? That's also something that I could help with. But as far as like actually collecting like an invasive sample from a human, that'd be way beyond.
No problem. Well, what's involved in culturing successfully? I know every bacteria is different, but what are some of the things you have to do? Do you put like a passivating nitrogen blanket over them, or how do you do it?
Yeah. No, you're pretty spot on. So we do work a lot with anoxic gases. So let's just start from ground one here. So what we'll typically do when we're making media in the lab, whether it's for aerobes or anaerobes, like starts with water and then some sort of nutrient, yeast extract, glucose, perhaps vitamins, there'll be other things, salt, that sort of stuff that would go in like a microbiological, the food that these bacteria are going to consume. And so where we differ from the aerobic microbiologists is we will start to make that media anoxic. We will start to get all of the oxygen out of it. And how we typically do that is by boiling the liquid. So we've got all our components in the media, sort of like a little primordial soup of goodies. And then we'll begin to boil the liquid so that way we can start to release some of those dissolved oxygens that are in, like say the water, for instance, in the media. And then kind of like you said, with nitrogen, at the same time, what we'll do is we'll flush into the flask that we're boiling an anoxic gas. It could be nitrogen, it could be C2, it could blend of the two of them. There's all sorts of anoxic gas mixtures that we could add, sort of depending on what needs to happen. And then, once we've got that media good and anoxic and we're confident in that, we'll then put the media into some specialized cultivation tubes. So if you think of like a traditional test tube, you pull the top off of it and kind of move the bacteria from tube 1 to tube 2. For the anaerobic stuff, we crimp seal them with like butyl rubber stoppers. And so we'll have like a butyl rubber stopper in the top of the tube, and we'll crimp an aluminum crimp seal on top of that. And again, we'll have a liquid in that tube that's not got any oxygen in it. And then the remaining headspace in that tube, in other words, the non-liquid portion, we can fill that with any gas that we want. So it's usually nitrogen, or it's nitrogen CO2, or there might be like a little bit of hydrogen in there. It just kind of depends on the need.
Gotcha. I don't know, is there like a bacteria that would be very useful and they can be cultured, but it just can't yet? Are there a lot of them like that, or only a few?
Oh, I'm sure that there are a lot of, there's probably some we don't even know about. But yeah, I would certainly say that there are ones out there that would be of importance to us as humans, but we just haven't grown them yet. Definitely.
And how do you know when you're sampling you've got the right bacteria? I would think there would be thousands of different things in a sample that could confound it. Fungi and yeast and protists, everything you can imagine.
Yeah, and you're not wrong, and that does happen sometimes. So yeah, what we'll typically do, a good workflow for, say identifying, if we're talking about like novel anaerobes, is we'll use some sort of screening methodology to sort of test for what the thing is that's growing on the plate. And so in our lab, we typically do that by screening. It's called 16S rRNA gene. And so that's a gene that is sort of very much a conserved genetic marker. It's found in all prokaryotes, and it's involved in their sort of development of the ribosome. It's essentially the small subunit on the ribosome that's a portion that they use for protein synthesis. And so what we do is we take that, some primers, and we'll do some PCR, and then we'll amplify that gene, and then we can compare that to various databases that have published data in them, and we can sort of see like, what do we have growing on this plate? That's sort of one methodology. Other labs use things like MALDI-TOF, mass spectrometry, and they'll blast the bacteria with lasers and then compare them to databases to try to figure out what they have. But yeah, we typically use some kind of a screening to see like what's going on.
We'll ask you to help identify. I mean, what are people looking for? What are you looking for? Sometimes it'll be, I guess, a new organism. What else would you look for? Like a metabolic pathway of a newly discovered organism? Like what are some of the things you check?
Yeah. So with respect to sort of how we check for that, again, typically what we'll do, whether it's bacteriological or mycological, whether we're talking bacteria or fungi in the lab, we'll take, say, like a fecal sample or something like that, or an enrichment from a fecal sample, and we'll extract the DNA from that. We'll run what would be called like an amplicon survey of that sample. And then we can get a breakdown of like, okay, here's a distribution of the abundances of the bacteria and the fungi that are in this sample. And then from there, we can look, say we have, 20 samples from beavers or blue whales or whatever the case might be. We can go back to a particular sample that was highly abundant in a particular type of taxa, a particular bacterium or a particular fungi we might be interested in growing because it's never been grown before. And then we'll start to enrich it in particular ways, this sample, to try to do what would be like a targeted cultivation effort of something very, very specific. And so in general, yeah, we're like fishing for new things. That's kind of what we like to do.
How hard is it? I mean, depending on the mixture of material you have, is it always like a multi-stage long process to do this, or what is it like?
Well, I'd say it's a mixture of a lot of things. It's definitely quite a few steps involved, and sometimes there's a little bit of luck involved too, right? A lot of what we do are sort of based off of differences in what would be called colony morphology. So for instance, what do these colonies, these dots, what do they look like on these plates? Because some of them may have a particular pigmentation, or they may be a particular shape, or there may be something, sort of a visible characteristic that we're using to screen them. And so sometimes it's just picking the right one. Sometimes it's using specific conditions, like we may have again, like say a targeted effort where we do something with the concentration of salt in them and make it so that organisms that can't tolerate a particular concentration of salt would like die, or will alter the incubation temperature such that, it's like really high or really low, or it's in a particular range. So there are variables that we can play with to try to hone in on specific types of taxa that may have like a good idea about what they kind of like to grow in and operate in.
You anaerobes form biofilms? Are there biofilms different from aerobes?
They can certainly be parts of biofilms. Yeah.
Any difference using the biofilms created by them? And it's a big question.
I don't know enough about biofilms to really tell you a good answer to that question, but I can tell you that there certainly are integral parts of biofilms that are made up from anaerobic bacteria, and we can find those anaerobes and biofilms, even in, like, say, the plaque on your teeth. They can hide in all kinds of places.
What about phages? See similar levels of phage activity in the aerobes versus anaerobes?
That's a great question, Richard. I honestly don't know. I do know that there are people that are interested in looking for phage and other sort of viral signatures in the genomes of bacteria, but I couldn't accurately speak to like the proportions that are in aerobes versus anaerobes.
But are there well-known phages for anaerobes as well, just like aerobic bacteria?
Well, what do you mean well-known phages?
Like the T4 phage, the bacteria phage. There's supposed to be a phage for every bacteria. So does that extend to anaerobes too? There should be associated phages with like every kind of bacteria you could find.
Oh, okay, I see what you're saying. Yeah, no, there are definitely phages that are specific to different types of anaerobes. Yes, a lot of phage. The answer is yes. There are definitely phage that are paired up with obligate anaerobes.
Okay, so same thing on the other side too here. Anything really unusual or unique to anaerobes you've discovered that does not show up in aerobes?
That does not show up in aerobe.
Yeah, besides a different respiration, is there anything that occurs on one side of the line that just never occurs the other side?
Richard, I'm not really sure. Nothing's coming to mind at the moment.
I'm there. I figured there probably wouldn't be. I just wondered if there was, So, well, I know I ask you questions all over the place. I know just because you have you study one part of an industry doesn't mean you know like everything, because there's like so many things to know is the same. But that's why I wanted to ask you just about some of the work you've done. So, are there any favorite anaerobic organisms you have for a certain reason? They do really cool things. They look really unusual. They're just strange.
Yeah, that's a really interesting question, and I think for me, I'll start off by saying totally biased response here, but probably my favorite would be like the first one that I ever found, and so that's a little Clostridium bacterium called Clostridium tanneri. It'll hold a special place that's sort of near and dear to me because it was a big part of my PhD project, and it was sort of my first foray in this anaerobic landscape. And so I think for me personally, it's that. For the rest of the lab here, it's going to be that the fungal AGF phylum, the Neocalipmastica mycota, and they are really, really weird in the sense that they're in a whole bunch of different animals, and they grow in such weird and wild pattern, and they do look really fascinating under the microscope. Yeah, they're very odd looking.
Okay, well, very good. Well, Sam, what's the best way for people to find out more about your research and to just learn about some of the microbes that you work with? Where can they go for a starting point?
Yeah, so I would probably direct them to our MCCF core facility page online. If you just Google Oklahoma State University Microbiomics and Culturomics core facility, that information will be there. I think all of the people in the core facility sort of have their LinkedIn or something like that, social media links sort of in that area in there. Yeah, people can find me online at LinkedIn or even on X under Microbe Miller. So I'll post periodic things, whether I'm out collecting samples, working with something in the lab. So if they want to follow along, that's great too.
Very good. Well, Sam, thanks for coming on, and I appreciate you fielding all the questions.
Yeah, well, Richard, again, thank you very much for having me. It's been a pleasure.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.